It's Dr. Stephanie, so we'll go for Dr. Gnanendra Shanmugam.
Okay.
Hello, is it audible
Hello.
now?
Yes, yes,
Yeah,
yes,
yeah,
yes.
this is Gnanendra Shanmugam here. Okay,
Okay, welcome, Dr. Dhamendra.
yeah, thank you.
Okay,
So let me share my screen first.
sure.
Yeah, so hope the screen is visible.
Yes,
Yeah,
yes. You're welcome, Dr.
okay.
Dhamendra.
So thank you for this opportunity. And unfortunately, my mic driver was not working. So just now I got fixed it. So, so basically my talk will be on cancer microbiome. So this is an area where one can understand how the microbes are playing a critical role in maintaining an human health, as well as how they are helping, how they are causing and diseases like cancer, and how they are helping in disease prognosis, and as well as how they are helping to treat the same diseases. So basically, I'm not an oncologist, I'm an bioinformatician. So in this lecture, I'll walk through what are the applications of microbiome and how we can do a complete analysis to understand and host microbe interactions and how they are helping us to treat the cancer diseases. It's not related only to a cancer. This can be applied to any kind of diseases. And as this conference is specifically on cancer, so I have chosen this as of a topic for today. So first, coming for an understanding what is a human microbiome. So we all know that we are being surviving with microbes in and around us, wherever the environment, whatever the environment might be. So we are always surrounded with microbes. So studying of that microbes is called as often microbiome, specifically in human microbiome. This is an prestigious project of NIH, National Institute of Health. So where they aim to identify what are the total microbes which has been surrounded by us, and what's the genome, and what's the impact, and what are the critical functions they have been playing, and how they are helping the human organism to survive. So if you come for the numbers of a microbiome, so if you see that we know that we are all the human beings is made up of 3.31 million genes, where we can say that when you compare and two human beings, we are having a 99.9% of similarity in the genome. Whereas when we look into the microbiome of two individuals, we have 80 to 90% of variation. So this is the one which decides that how an human being will function, how an human being will respond to an specific diseases and to an environmental factors. So overall, we can say that we are having an almost 10,000 bacterial species which has been surrounded by us, and where it accounts to an 10 to 100 trillion cells if you're going to count the total cells which has been present in a human, including our microbes. And it can be said as of an unique ID code like one nature, like our fingerprints. The microbiome will also be unique for each and every individual. So in future, based on this, we are going to have and specific medicines like treatments. Various treatments can be given based upon the microbiome by taking a microbiome profile of each and every individual. At current, the applications has been going on. So in which specifically we will be looking into an gut microbiota. So that's called as of a place where the critical role has been playing. So we have an n numerous of an bacteria which is playing a role like specific functions like maintaining a nervous systems, breakdown of food components, and responding to an pathogens, promotion of fat storage, and immune system stimulation. That's the place where we are going to concentrate more as it's related to an cancer, and how the vitamins has been biosynthesized. So all the specific roles has been maintained by the bacteria which has been present in our gut. So the gut microbiome has gained and more attention, and this is known as often other genome of a human. So which has been published in Nature by saying that it's our other human genome. So it's a catalog of our body, so which maintains how a specific individual will perform and how the disease prognosis will take place when there is an changes in the microbiome of an gut. So based on that, we have in different parts of the body. So where we have in different kind of microbial organisms which has been found, or we can call it as of in various communities which has been present. So if you look for an example, an oral microbiome, so which will be helpful for us to study and periodontal diseases, gingivitis and caries, as well as placenta microbiome, so which can use for studying in different kinds of diseases, and vaginal microbiome, skin microbiome, and gut microbiome, so which is an very important for each and every one. So like maintaining the body, so it helps us to get rid of many diseases like metabolic syndromes, diabetes, and colorectal cancers, IBD, psychiatric disorders, so and so. So by understanding that what kind of microbiome has been present and what's the nature of this microbiome and what kind of functions this specific microbiomes has been performing, so one can understand how they have been interacting with their specific host and how they are helping the host or how they are causing the diseases. So coming for the maintaining of that, where we are having a very vast environmental factors which decides the gut microbiota, like host genetics and the exercise nature, the habitat, the habitual of the person, and even stress, antibiotics, and age-related microbiome are there, gastric mobilities, gastric acid secretion, and diet, mode of delivery. So which is very important that based on what kind of birth we are having, like uh, a natural birth or an C-section birth or in geographical location. So these are the various parameters which decides how an microbiome should be and what are the microbiome and what is the gut microbiome of an each and every individual. So if you look at this, we have the dynamics of microbiome will be keep on changing as soon as the human is growing on, and to when we are shifting to another locations, what are the microbes which has been surrounding us. It depends upon the environment, what are the food habits which we are taking. So this is an example like which shows that at the birth we have only one Enterobacteriaceae family. So where the baby is being attained with only Enterobacteriaceae family, where as soon as at an 1 month birth, after 1 month you can see that there is an increase in the microbial diversity, where we have a number of families of bacteria has been seen. So as soon as the baby is growing, we can see that the diversity has been increasing and inter-individual variability will be decreasing, which says that based upon the functions which we are going to perform, we need a specific kind of bacteria which can give us an energy for further development of the human body. So this and another example like which shows you that how the food habits can change the diversity in the gut. So once if we have a high microbial diversity, which is helpful for enrichment of high metabolic functions, as soon as if you're going to change to an Western diets like pizza, burgers, whatever the food which we are going to use, so it depends upon it will definitely it will have some impact on our gut microbiome. So where the microbial diversity will be depleted and which can result in decreased metabolic functions. So maintaining a gut microbiome is very essential. So if this is an example like which can clearly explains you that how the diet has been playing a role. So you can see here non-Western diets like rich foods can increase the microbial diversity in gut microbiota, like bifidobacteria, bacteroids, prevotella will be increased, and there will be a decrease in firmicutes. So in insignificance, we can see that side chain fatty acids can be increased, like a butyrate, acetate, and propionate, which can improve the mood. So mood of a person can be dependent upon the food which we are going to take. Whereas if you see in Western diets where there will be a decrease in the microbial diversity like bifidobacteria, bacteroids, and prevotella, which in result decreases the side chain fatty acids like butyrates, acetates, and propionate, which can bring an individual to a different state of nature. So this is how the gut microbiota has been playing a role. So understanding that how the microbiome has been distributed throughout the body. So we have numerous studies like where we are going to study in human microbiome composition. So which gives you a clear idea that what are the microbial community which has been seen in gastrointestinal variation and urogenital variations, and you can see that oral variations and skin microbiota. So there is a distribution of microorganisms which has been seen at different places. So once we can understand that what kind of bacteria has been occupied at specific regions, and we can know that what are the functions has been performed by this specific role of bacteria. So once we can understand that, we can say how they have been helping or how they have been causing and diseases, and how one can overcome these diseases by making and specific changes in a microbiota. So this is another example where we can see that when the baby has been born, we can see that if the baby has been attaining a good bacteria, so the health status will be very good. Whereas if the baby has been attaining and bad bacteria, so there will be an disease status. So here there is and one research study which says that if the baby has been born through an C-section, those babies will be more prone to an asthma-like conditions, whereas the baby born in a natural condition will be having an good bacteria which can give an healthy status to the specific individual. So this is an example where it leads to an breakdown of our epithelial cells and uptake of various kind of bacteria, so which can lead to an different kind of studies, different kind of diseases. For example, here we are going to deal with often disc biopsies where it can go for an IBD diseases, so which can lead to an colorectal cancer. So with reference to an carcinogenesis, we can see here that symbiosis and dysbiosis, so which can lead to an tumor suppressive effects as well as oncogenic effects. So if there is a symbiosis in your microbiome, then it helps in anti-inflammations and barrier functions and antioxidants by helping with the help of an specific functions. Whereas if there is an dysbiosis, then we can see an oncogenic effects like inflammations, gut permeability, barrier failures, like DNA damage, which is responsible for most of the cancers, and estrogen metabolism and signaling pathways, which leads to an uh, cell proliferations. So in specific to that, we have an microbiota where dysbiosis can influence an anti-cancer immune responses and stimulate chronic inflammations, and as well as it helps in producing and cancer cells. So produce microbial genotoxins which inflicts and DNA damage. So these are the microbiota which can cause an which can produce an genotoxins which can cause an DNA damage in sense of mutations, whatever the thing we are calling, so which leads to an cancer. And as well as the immune system are the one which will be affected based upon the microbiota, so which can decrease the antigen presentation and T cell mediated responses and abundance and diversity of B cells and nuclear factors and immune tolerance leading to chronic inflammation. So this is an interrelation between the immune system, microbiota, and the occurrence of cancer genesis. So this is an example like which shows you that how the bacteria can influence an colon cancer. So where the bacteria can cross the epithelial membrane and where it can activate an macrophages, which can lead to an epithelial cell proliferation by activating the STAT and nuclear factors, so which can lead to an ROS factors like reactive oxygen species will be produced more, so which can lead to an DNA damages and which can lead to an chronic inflammations, or we can call it, it can lead to an adenoma and colon cancer. So through the production of primary bile acids, secondary bile acids, and peptides or amino acids, and by producing an genotoxins, so which can directly influence the carcinogenesis. So one should understand that what is the role of each and every bacteria and why we should maintain an healthy gut microbiome in order to protect ourselves. So this is a study where we are going to use, and this is the study which we call it as of a microbiome. So where we are going to sequence the entire genome of a microbiome seen in a specific individual. So by using your 16S rRNA sequencing, so which is an markers for a bacterial species, and 18S RNA sequencing for fungi. So in this microbiome, so we are going to study that what kind of bacteria has been present and what is the role of that bacteria and how we are going to do it. So how and what are the functions which has been maintained by that bacteria. So this is a simple layout that which says you that how we are going to perform these studies. By taking a sample, we are going for an 16S RNA sequencing of that bacteria, and we are going to classify that to which kind of to which family this bacteria belongs to, and what is the diversity in this specific bacteria, whose population is increased, whose population is decreased in a healthy state, and as well as in a disease state, say example like cancer studies. So we know that 16S rRNA gene is very essential, is used as of an markers, and its main advantage is like we have an, it's used for identifying and bacterial taxonomy, and it can be used for constructing and phylogeny to know that how far the bacteria have been interrelated with each other. So where we're having an 9 hyper variable regions which are used for sequence identify and specific different kinds of bacteria. And this is the strategy of MiSeq technologies, or high throughput screening, or next generation sequencing, whatever the name we name it. So these are the variable regions which will be found in which can be used for sequencing your 16S rRNA. So we have an V1 to V3 regions where we are going to use an Roche 454 machines to identify the specific bacterial community by using this V3, V4, V1, V3 regions. And next one is often more predominantly used V3, V4 regions by using an MiSeq technology, and V3 to V5 by using an Roche 454, and HiSeq for V4 regions, V6 to V9 regions, again Roche 454. And for full length like V1 to V9 regions, we are going to use an PacBio system. So this is an methodology, this is an next generation sequencing technologies which helps us to sequence the entire microbiome of any sample, and we can study that how the bacteria has been present. So these are the primers which has been used for sequencing. Like say for an example, if I want to specifically check this V3 to V4 regions, then we are going to use an primer 337F, and V4 region like A0, 805, or that's an reverse primer. So this is the region which primers which we are going to use to amplify that specific set of regions. And once we get that, we are going to delineate that what is the genus which has been seen in that, and we are going to relate that how the specific genus, what is the role of the specific genus, what kind of functions this specific bacteria has been performing, and how it has been helping to cause the disease, or how it can help individual to overcome a specific disease. So here we are going to study about an population genetics, like saying that what are the specific bacteria has been seen in an samples like alpha diversity and beta diversity, which can explain you the richness and evenness of the sample of the bacteria. So where the richness says that how many microbes have been present, and the diversity says that how the microbes balance to each other. So what is the specific number of bacteria and what is the function? So whether the richness has been seen equally, whether the richness is there or evenness has been maintained in the samples. So we are going to check that. And beta diversity is like making a determination between two different kinds of samples like treatment A and treatment B. We are going to analyze that how far the two samples looks like, and what are the different kinds of bacterial abundance has been increased or decreased. We are going to study in that sense, and this is how we are going to prepare the data, like identifying an OTUs, like so how many number of OTUs has been present, and how far the two samples has been clustered with each other by using a different functions like a PCO analysis, which can explain you that how far the samples has been seen close to each other, or they have been seen apart. So which can give you a clear identification that whose bacterial diversity has been increased or whose diversity has been decreased. And once we have this specific data, we can easily identify that, we can easily come for a conclusion that what is the function of each and every bacteria. So this is a basic workflow, so where we use a specific tool called as of an CHIME for analyzing this a microbiome study. So here we are going to collect an microbial samples, and then we are going to use an 16S RNA approaches as the methods which I explained in the previous slides, like next generation sequencing. So once we get an 16S RNA sequencing, we are going to group that into an specific OTUs, and we are going to classify that what are the different kinds of an OTUs has been present, and then we are going to identify that bacteria by using a different bacterial databases like Green Genes or NCBI or RDP or Silva databases. Once we have this specific bacterial composition, then we are going to make a relation that whose abundance has been increased or decreased, and then we are going to construct even an SNP studies can be done, and then we can go for an functional abundance and functional studies, which can give you a clear idea that what is the role of each and every bacteria. So this is a slide which explains that how one can use this specific technology to treat the cancer patients, like identifying what is the microbiome which has been seen in a healthy individual. By using an high throughput sequencing, we are going to study that what are the composition of each and every bacteria in a healthy individual, whereas in cancer individual, we are going to study what is the composition of each and every bacteria. And then we are going to make a comparative analysis like whose functions has been increased and whose functions has been, what kind of functions has increased, what kind of functions has decreased. And then we are going to make an system biology studies like which has been interlinked with each other, what function has been interlinked with each other, and whose function has to be increased or decreased. And then we are going to make medicines, either in the form of a diet or bacteriophages or probiotics or prebiotics or even an antibiotics can be used based upon the nature of the disease and how role of the bacteria. And one can use this specific diet to get back a cancer condition to a normal healthy condition. So that's a fin study. So where we have and different kinds of an applications like fecal microbiota transplantation, prebiotics and probiotics and postbiotics, which are successfully used to increase the diversity of an gut in an individual. And this is another study where it says that how we can use this specific microbiome study to bring back to an healthy state of a diseased individual. Like we have and three different kinds of treatment options. Like first generation is like directly taking an fecal microbiota by understanding that what is the frequency of each and every bacteria, and we need to decide the donor. So whether the donor is of an healthy donor, whether he belongs to the same family or not. So what is the different kinds of roles which has been performed by that microbiome. So we are going to collect those healthy microbiota from a donor, and then that will be given as of an FMT to a disease state to bring back an healthy state. Or we can go for an precision replacements, like a specific kind of bacteria whose functions has to be increased, and it's called as of an synthetic microbiome. So where we can choose a specific microbial bacteria which can perform in specific tasks, and then we can make an synthetic cocktails to bring back an healthy state. Or the next one, or the third option is like targeted functions, like what is the role of each and every bacteria and what kind of molecules are like secondary metabolites, what are the bacteria has been producing in order to improve the health of a specific individual. We can identify those specific molecules, and this can be used as often drugs. So this is an options which has been available to treat the cancer by using that. And this is the success story of an fecal microbial transplanter, so which has been successfully used to treat the cancers in a different patients. So this is all about the microbiome research, and this often gist of the microbiome. So what we know till now is like we have 25 phyla and 2000 genera and 5000 species has been present, and 80% of that metagenome has been mappable, and which account for 316 million genes. And still what we know is still there are undetected bacteria, and it says that the research has been saying that there are hidden taxons and strain level diversity. So where 20% of sequences are not matching to the microbial genome. So it means that we have in some other bacterial species, or maybe some errors in the sequencing, or and 40% of the functional genes are not known to the functional databases. So once we can explore all this, then we can use this microbiome research to successfully treat the cancer diseases. So thank you for this opportunity. I'm happy to take the questions if any participants is having any doubts.
Thank you, Dr. Gnanendra, for the nice presentation about the linkage between cancer and microbiome, and the interesting study that we are going to conduct with regarding or focusing on identifying individual bacteria and their function between healthy and cancer individual. Thank you for alerting us on that. So
thank you.
Anyone with a question, please may ask Dr. Gnanendra regarding the microbiome and cancer. You're welcome. Anybody with a question to Dr. Gnanendra?
Yeah. Hello.
Hello.
Yes, you're welcome.
Yes. My name is Benson, and thank you for the nice presentation, Gnanendra.
Yeah, thank you.
I have a question. Can you maybe just touch on the issue of microbiota and the extracellular vesicles in relation to cancer?
To be honest, I'm not deep into the cancer research, but I
Yes.
just gave a gist of the microbiome.
Okay.
So how it can be used for cancer
Okay.
research.
Okay.
So
Okay.
maybe I need to do some more research on this to study how specifically they have been helping in the intracellular level.
Oh yeah. I mean, okay. Thank you. Thank you. Thank you. I understand. Yeah. Yeah.
Yeah.
Can I
Thank
ask
you.
a question?
Yeah. Yeah.
Thank you, thank you.
Thank you.
Ben, thank you, Dr. Narendra. Anybody with another question? Any more question?
Yes, I have a question, please.
Yes, you're welcome, Dr. Lamis.
What is about the link between the microbiota and the nutritional support in cancer patients?
Pardon, can you come again?
Dr. Lamis?
Yes, what is the link between the microbiota as a causative agent in cancer and the link of it with nutritional support?
Link between cancer and microbiome?
Cancer microbiota and nutritional support of cancer patients.
Yeah. Okay. So you say for an example, like if the fiber diets has been taken by a specific individual, the dysbiosis cannot be happened. So which can prevent the inflammations of your colon, so that there will not be any crossover of the bacteria into the intestine. So, for example, we have an specific bacteria like H. pylori. As far as I remember, that's the one which is very crucial for colon cancer. So, which needs to cross your which will cause an inflammations. So, this can be prevented by having an nutritious food.
Okay, thank you, Dr.
Okay, nice
Narendra.
answer.
Pardon?
Thank you, Dr. Granedra.
Yeah,
I
thank
think
you.
Dr. Lamis is satisfied. Maybe in addition to what you are saying, probably apart from the direct effect of the dysbiosis of the microbiota, probably there is a link between cancer epigenetics with the products of bad bacteria in the gut. Probably there is this kind of an association that can lead to cancer also.
I couldn't get the question, please.
It's not